Okay, thank you everyone. So I make that 1:05, so we might get started now. Thank you for coming along to lecture 2 of the Fundamentals of Fluorescence Microscopy series. My name is Paul McMillan. I'm the manager of the Biological Optical Microscopy platform at the University of Melbourne. Before we get started with today's webinar, I'd just like to acknowledge the traditional owners of the lands on which we're meeting, which in the Parkville campus is the Wurundjeri Woiwurrung people of the Kulin Nations. I'd like to pay my respects to their elders, past, present, and emerging, and welcome any First Nations people with us in the meeting today. This webinar series is being run in conjunction with Light Microscopy Australia. If light microscopy or fluorescence microscopy is a large part of your project, then I'd encourage you to sign up. So it's part of the Australian Microscopy and Microanalysis Society. Let me just put my laser pointer to good use. Yeah, so part of the Australian Microscopy and Microanalysis Society. We've got 250 members across Australia. For your membership fees, you get access to travel bursaries to attend conferences, workshops, and lab visits. We also run fairly regular data analysis workshops across Australia. There's an email of upcoming events, many of which are online, like this one, so that people from across Australia can access. And then finally, there's an annual image competition, which is open to anybody based in Australia to submit your images and win some cash prizes. The next big event for light microscopy that's coming up is the Australian Conference on Microscopy and Microanalysis, which is in Sydney in February next year. And that will have 4 days of scientific talks along with a couple of days of workshops before and after the conference. That will incorporate not just Light Microscopy Australia, but also the Australian Bioimage Analysis group as well. And the website is there if you're interested to look at that. And if you want to join LMA or AMMS, then you can go to microscopy.org.au. All right, I'm just going to turn off my video camera so you guys can concentrate on the slides rather than me. Yeah, so as I say, this is lecture 2 of a 3-lecture series. So last week we went through the fundamentals of fluorescence, fluorescent dyes, proteins, tags, and also fluorescence microscopy and the structures within the microscope that are important. Today's lecture will cover sample preparations, some of the experimental applications that are possible on fluorescence microscopes, and then we'll touch a little bit on image ethics as well. And then next week, my colleague Ellie Cho will cover the fundamentals of bioimage analysis. So if you are generating fluorescence microscopy data, then that's a very useful one for you to know what to do with the data once you've generated it. All right, there we go. All right, so lecture 2, as I said, this is split into 3 parts. So we'll start off with sample preparation, and here I'm going to give very general advice on sample preparation. So there's a wide range of samples that are possible to use on the microscope, so it's very difficult to give direct advice. Experimental applications, we'll be focusing primarily on the applications that are available within our platform. Although those are more widely available if you have the relevant equipment. And then finally, we'll go through a little bit in terms of image ethics. So once you've generated your data, what should you be doing to it? What's ethical, what's not ethical? I would say if you have any questions at any point, feel free to put it in the Q&A section of the webinar, and I'm happy to answer those at the end. All right, so sample preparation, as I said, I wanted to go through some very general sample prep rules. We covered some of this in the first lecture, but it's good to reiterate and go back through this. So in general, your best imaging is going to occur as close to the cover slip as possible. That means that it's best if you can actually mount your sample onto the cover slip rather than onto the slide. You're going to get the best imaging quality on the cover slip interface. This is especially important if you're doing three-dimensional imaging as well. As we covered in the first lecture, your cover slip should be 170 microns thick. That's a number 1.5 cover slip. Again, you can check on the objective you're using if that is the expected thickness that the objective wants. One other thing we covered during the initial lecture was to select the best fluorophore for your experiment. So, we're looking at things in terms of, what the wavelengths for excitation and emission are, what the photobleaching characteristics of that are. Again, going back to excitation emission, try and match those fluorophores to the microscope setup, or vice versa. You want to be imaging as efficiently as possible. One thing I'm always keen for our researchers to do is to titrate the concentrations. So just because you buy a dye from a vendor and it says to use it at a set concentration, that's not always the optimal thing to do for your sample. So I would strongly suggest that you try and optimize that as much as possible. So if you're doing transfections, titrate the DNA concentrations. If you're doing experiments where you're fixing samples, then, fix the concentrations and the ratios that you have of each of the fixatives. Same for antibodies or stain concentrations. Try and optimize those as much as possible. And again, really important, this is still a scientific experiment that we're performing, so you need positive and negative controls within your experiments. Over here on this panel here, we've got image taken with slightly different cover slip thicknesses. So here, and here you can see the image quality starting to deteriorate as our cover slip thickness gets thicker. Other thing that you want to do is to try and fix your sample, if you are fixing it, as gently as possible. So you don't want to do your fixation which is too harsh and therefore potentially putting in errors into your sample, causing more problems than you need. The other good habit to get into is to image samples as closely to preparation as possible. Again, your fluorophores can die off over time, and you're changing the intensity that you could possibly be getting out of the microscope depending on when you acquired the data. This is one that we see quite a lot, is that people don't necessarily use this correct sample mounting for the microscope. So here we're using an oil immersion lens with a refractive index, normally 1.517. Here, if we look in 3D, if we're using water, or mounting the sample in water versus mounting it in something with a matching refractive index, you can see the difference in image quality. So here we're matching the correct refractive index. We're getting nice 3D data. For mounting it in water, that's causing a deterioration just because we have a mismatch between the oil immersion and what your sample is mounted in. The other issue that we often see is people mounting with maybe a commercially available mountant that has DAPI included within the mounting media. We normally don't recommend that. Often the quality of staining is not very good for the DAPI channel, and it can also cause high backgrounds as well. If you remember from the first lecture, that DAPI goes all the way into your green channel. We can get problems there with extra background signal coming from that DAPI. So please, if you're going to stain DAPI, please do it as one of your staining steps. Wash out any unbound and then mount with a mounting media without any DAPI included in. All right, next thing we're going to go through again are some specific guidelines around, this live versus fixed. What are the types of things that we need to think about? So if we've got our cells or tissue and we want to image them live, then that's probably going to limit the way we can stain our samples to either fluorescent proteins or things like cell permeable dyes. If we're happy to fix our samples, then that opens us up a little bit more. So we can still use fluorescent proteins and dyes, but we can often then use antibodies as well to go in and identify specific proteins. So we're going to go through some things to consider for live cell imaging, and then we'll go through some of the steps required for fixed sample imaging. All right, so with live cell imaging, Seems fairly obvious, but we need to keep our samples alive. There's a few ways that we can do that. So the first thing is to match the temperature that's expected for those samples. So if you are, culturing mammalian cells, for example, then you want to incubate the microscope up to 37 degrees. In the picture here, you can see a microscope with this Perspex box around it that allows us to warm up that environment. The other thing that this system often comes with is control of the gaseous environment. So you'll see there's a heater here, but also a controller down here for controlling concentrations of different gases. The most common one is CO2. That is there to maintain the pH of the media which your sample is living in. So you don't want that pH changing over time. That's obviously going to change the behavior of your samples. Humidity is also another important thing. So we want the media that the samples are sitting in to remain. So anything that we're flowing on, in this case CO2, needs to go through a little bubbler system. So there's a zoomed in view here. So anything that we're blowing on top of the samples on the stage here is actually humidified, so stopping any evaporation. Often in our platform, we use gas permeable seals on the top of samples to allow the gas to come in, but stop evaporation from going out. And then the final thing, I've called it food, but obviously we need to provide the sample with nutrients, right? So you need to provide it with a buffer that is going to provide it with all the nutrients it needs for your imaging experiment. So you don't want to be imaging your live cells in PBS, for example. Some of the other things in terms of microscope setup to think about is the, first of all, the microscope that you're using. So again, this can come down to availability. So some of the instruments you might consider is like a spinning disc confocal system, which can be fairly fast acquisition, but it's also a little bit more gentle on the samples. If your confocal is fitted with a resonance scanner, this will scan the laser across your sample much faster than a traditional galvo scanner. That is also a way of being more gentle on the sample. If you have an AEST system, then you can use the AEST fast or the multiplex modes, again, to sweep across multiple lines of pixels at a time. Again, that's going to be a much faster and gentler acquisition on your sample than a standard confocal system. And if your samples are really delicate, then you might want to go to a lattice light sheet system where being as gentle as possible, we're only illuminating what we're imaging on that microscope. So this is probably at the best in terms of sensitive samples. Next thing we need to look at is selecting the correct fluorophores. So some of the things we're looking at here are, we want them to be as bright as possible. So we're putting a minimum amount of light onto there. You could also look at detector sensitivity as well here to make sure, you're being as efficient as possible in collecting whatever photons are emitted. For this, what we normally recommend is to use longer excitation wavelengths. So try and avoid ultraviolet light. When we go out in strong sunlight, we have to put our sunscreen on. It's the same idea for the cells. We don't want excess ultraviolet light potentially damaging the samples that we're trying to look at. So yeah, HOST is probably not the best dye for doing long-term live cell experiments on. Here, also reducing phototoxicity. So again, those long wavelengths are better, and low excitation levels are good as well to try and reduce any damage that we're causing to the samples while we're acquiring that data. And then the last thing, as we discussed before, is to, again, image through that cover slip thickness or through an optical plastic. We often suggest these IBIDI chambers and dishes for our researchers. They have a cover slip thickness base. So you can get a high magnification lens there. You're going to get a high image quality because you've got the correct thickness, correct refractive index going through that sample. You can purchase something like a Sykes-Moore chamber. You can now 3D print these types of things as well. So these dishes can be quite expensive, In this setup, the cover slip is the thing that you're swapping in and out, and this is being reused between your experiments. So that's some of the things to consider just generally in terms of live cell imaging. In terms of fixed sample experiments, we'll go through a few of the different options here. So what we're doing when we're performing sample fixation is essentially chemical cross-linking of the proteins within your samples. There are a few here which are kind of commonly used fixations with some examples on the image on the right-hand side there. So glutaraldehyde, for example, will give you strong fixation, good morphology, but often results in poor stainings if you're using antibody-based staining. Paraformaldehyde, for example, will give you weak fixation, poor morphology, but allows you to have good stains. So often these are used in combination. And again, we often tell researchers if they're new to their sample, then go and hit the books and see what's been published before in terms of sample prep reagents and concentrations. Acetone methanol is one that works specifically for different types of samples, but often that can cause too many artifacts. There was more recently this alternative called glyoxal, which is seen as being kind of a middle ground between your glutaraldehyde and your paraformaldehyde. Again, depending on the structures that you're trying to stain, which fixation you use will change depending on those structures and those samples that you're trying to visualize. All right. So what I'm going to do now is go through some different generalized workflows for fixing different samples. So in this case, we're looking at cells. So an immunofluorescence assay from, perhaps maybe a monolayer of cells. So I mentioned your best imaging is going to happen on the cover slip. So it's best to grow or attach your cells directly to the cover slip. If you had non-adherent cells, then you can try and attach them to the cover slip using something like a polyal lysine, or CellTac is a commercially available reagent which will do the same thing. Once your cells are attached, obviously we want to fix those, cross-link those. We've covered some of those potential reagents for doing that in the previous slide. Again, the key thing is to titrate the concentrations and the conditions, so length of time, temperature, that type of thing. Again, that will vary very, very differently depending on the structures that you're trying to stain and visualize. Once we've fixed everything, what we want to do is to permeabilize the cells. So here we're punching little holes in the membranes to allow reagents in and out of the samples. So obviously you need to fix before you do that. One example of this is Triton X100. Here again, you're best to try and titrate concentrations here and length of time of incubations, things like that. And again, different agents will work differently for different organelles. So again, time to hit what's been published in the past and try and use that as a starting point for your analysis. As we're doing an immunofluorescence assay, we're going to come in with antibodies. So first thing we want to do is to block. So normally what we're doing is using like a bovine serum albumin or a serum from a secondary antibody species. In this case, we're trying to reduce the non-specific antibody binding. So we're putting high protein concentrations on to make sure that any antibodies that are binding specifically to the protein targets that we're interested in. After that, we'll go through antibody labeling and mounting the sample, and we'll put that on the microscope, and we'll cover the antibody labeling side of things in a few slides' time. So that was kind of cell-based immunofluorescence assays. The next thing that we're going to look at is tissue section experiments. And in this case, we are going to paraffin embed the tissue samples. So there's a few steps here. Before we go through those, I'd just like to highlight at the University of Melbourne, we have a histology facility which will train you how to do this. I can also run fee-for-service experiments to do this for you on your behalf. So feel free to reach out to people, histology team, through that link at the bottom there. In these experiments, we will isolate the tissue from the source. Here we're going to, again, chemically fix that sample. So through something like a paraformaldehyde, we're going to embed that sample. So dehydrate and embed the sample in the paraffin wax. We'll then take that paraffin block to something like this, which is a microtome, and we can cut very thin sections of that tissue sample. This goes back through, so it goes onto a slide, back through rehydration. Then we have antigen recovery, so either heat or enzymatic recovery of the antigens, making them available again to the antibodies to bind. And then we go through, again, the block and antibody labeling steps, and we'll go through those in the next few slides. So that's paraffin embedding. There is another type of sample prep for tissue sections, which is cryopreservation, which again, we'll isolate the tissue. We go through the chemical fixation step with PFA or neutral buffer formalin. We will infiltrate the sample with sucrose, which provides cryoprotection. We'll go through optical cutting temperature embedding. So cryogenic freezing using the OCT, so very, very quickly snap freezing. In this case, we need to section it using a cryostat. Here we can control the internal temperature, so they're still maintained at a lower temperature for this, and cut the sections. Put them onto a slide and air dry onto the slide. If you want to store them for long periods, then that can be done at -80 degrees, before again going back through rehydration, blocking, and antibody labeling again and again. If you're interested in cryo-sectioning samples, then the University of Melbourne histology facility is available to assist with that. All right, so I mentioned antibody labeling as one of the key outputs in all of those applications. So in this case, what I'm going to go through is what we call an indirect immunofluorescence assay. I think I touched on it in lecture 1 a little bit. In this case, we've got our antigen, our protein that we're interested in here. It's sitting on a piece of tissue. So this is our protein that we want to label, sticking out of the tissue. We have a primary antibody here, which recognizes our protein that we're interested in. And then we have a secondary antibody here, which binds to this primary antibody. And it's the secondary antibody that has the fluorophore attached. Again, it's not shown in this figure, but here we can have multiple secondary antibodies binding to the primary, and that gives us an amplification in terms of intensity coming off the sample. All right, so what are the things to consider when we're performing antibody labeling? So for the primary antibody label, obviously it's got to be specific for the protein that we're interested in. One thing that we need to be aware of is the animal that the primary antibody was raised in. So in this case, we're saying rabbit. So in the table down here, we've got an anti-GFP. So this is raised in mouse. So it's a mouse anti-GFP antibody. So this goes on after we've done the blocking step, obviously is incubated for a period of time. We want to go through multiple washes there. Again, we want to wash off any non-specific binding there. We only want those antibodies still attached to the protein that we're interested in. After that, we can go in with the secondary antibody label. And what we want to do is match the antibody used in the primary experiment, right? So in this case, the primary for GFP is a mouse anti-GFP. We're going to go in with a secondary antibody here, which is raised in goat, but it's an anti-mouse antibody. So it's going to detect this mouse antibody. So this is the goat anti-mouse. It's going to detect that mouse antibody. And in this case, it's conjugated with an Alexa fluorophore fluorophore. Okay, so we just need to be careful about the organisms, the animals that these antibodies have been raised in. Again, once we put the secondary label in there, we're going to wash thoroughly to make sure that we have taken off as much of that of those antibodies as possible. Again, we want to reduce non-specific binding. There is an optional step here where you can go back in and fix those samples again. So again, you're chemically cross-linking those antibodies together and to the sample. That can sometimes help if you're finding that your signal is disappearing. And finally, you would mount that sample again, slide and cover slip imaging on the microscope. So if we come down to the table again, so we've got our anti-GFP, we've already kind of walked through, that's an anti-GFP antibody raised in mouse. In the secondary, we've got an anti-mouse antibody that's been raised in goat, and that has the elixir fluor 488 attached. For our co-stain in this example, maybe we're staining actin, so it's an anti-actin antibody that's raised in rabbits. And in this case, we've got an anti-rabbit antibody, which has been raised in sheep, and that has an elixir fluor 568 associated with. So we don't have overlap of any of the species within this table. Okay, so that's everything I wanted to cover on sample preparation. If you give me 2 seconds, I will try and find polls and quizzes so we can set that up. And I will launch that and give you guys a couple of minutes to go through and try and answer some of those questions, just to test your understanding of what we've covered so far in the lecture. Okay, so for question number 1, the question is, what is the best choice of cover glass for most confocal microscope applications? In this case, it is the one that 88% of you selected, which is number 1.5 cover slips or 170 micron thickness, which is good to see. The second question is, what should you take care when choosing an antibody panel? The correct answer here is that your secondary antibody should target the species that your primary antibody was raised in. So that's the second question there. And again, a large proportion of you guys got that answer correct, which is good to see. Okay, so let me just close that down. We'll go back to the presentation. Okay, so yeah, that was some kind of very high-level general guidelines on sample preparations and what to think about. Next, we're going to go into experimental applications, and we're going to spend quite a bit of time going through possibilities in this space. And there are a number of different techniques that are available. I've chosen to divide it up based on size of sample. So here we can go from kind of model organism side of things, so like fish and flies, whole bits of tissue from model organisms like mice, all the way down through tissues into cells. And then this red line here is actually what we call the diffraction limit. So this is at about 250 nanometers of wavelength. And this is for standard fluorescence microscopes, kind of the resolution limit. So because of the wavelength of light we're using, we're collecting in the kind of 500 nanometer ranges. Our resolution is limited to about half of that. So yeah, for standard microscopes can only see structures if they are greater than 250 nanometers apart. If we want to go below that, then we start moving into this green sphere area here, which is in the super resolution microscopy domain. Again, that will take us down to probably about 10 to 20 nanometers. There's a few very specialist techniques which can go much, much lower than that now. Certainly we don't have those available within the platform. And yeah, if you need to start going down to atomic levels, then we need to start talking about electron microscopy, and that's not something we're going to cover in today's presentations. Okay, so we're going to start at the top end, at the kind of high sample side, and we're going to cover a few applications. So light sheet microscopy, multiphoton microscopy, slide scanning, high content imaging, and wide field microscopy. And we'll go through a few slides and give examples of how these work and examples of some of the data that's been generated. So light sheet microscopy is primarily used in developmental biology and neuroscience. It is a little limited in resolution in that will only give you cellular resolution. Again, there are lots of different flavors of light sheet microscopy. Here we're focusing primarily on large sample light sheet microscopy. As the name suggests, the sample is illuminated by a light sheet. So we have cylindrical lenses on this side and this side, which create what we call a Gaussian beam. Parallel to the beam, which is our excitation light, our sample is sitting here. Parallel to that is our detection arm. So we have an objective here and a camera going off into the distance. So here we're only illuminating our sample, which is in focus, so a single sheet of light going through in focus with the objective. We can then move the sample. In this case, it's showing a rotation within the sheet of light to build up a 3D data set. There are systems that will also translate the sample up and down through the beam. In this case, we're primarily using a dipping lens. So this little cuvette, as is drawn here, is often filled with some form of solution. And ideally, for this to work, the sample needs to be optically clear. So this is why it's primarily used in developmental biology. Things like Drosophila embryos, zebrafish embryos, and worms are fairly optically cleared. If you want to put larger pieces of tissue in there, then you need to chemically clear that tissue out, and that's something that we do at the university quite a bit. When it comes to tissue clearing, this has been around from a very, very long time, but can involve some fairly nasty chemicals. There are two main groups of methods that are used. So the first one is organic solvents. In this case, we're dehydrating the sample again, as we did for the tissue sections previously. And what we're trying to do is match the refractive index of the tissue with an organic solvent. So in this case, there's one approach here. This actually comes up under aqueous-based. This is the clarity approach. You can see a brain here before and after clearing. So you can see it's almost completely see-through. With the organic solvents, these are not always applicable to fluorescence proteins. So often the chemicals used will damage the fluorescent proteins and get rid of any fluorescence. Some that you may have heard of are things like BAB, disco, ethyl cinnamate is one. So a lot of our researchers use the disco-based techniques. And then finally, image in ethyl cinnamate, which is slightly less toxic than some of the other chemicals. Some of the other clearing methods that are available are more aqueous-based. So we can have removal of lipids. So systems like Clarity will do that, again, followed by refractive index matching with higher refractive index solutions. So yeah, the approaches here are Clarity, Cubic, CDB, and Switch. The majority of researchers, as I say, at the University of Melbourne are using the disco-based techniques. Benefit is it makes the sample slightly smaller, and the sample is fairly hard and easy to manipulate onto and off the microscope. But yeah, the instrument itself can handle any of those techniques. One of the downsides here is it can be quite tricky and difficult to set up and quite expensive depending on which of the clearing approaches you decide to take. But again, we can help put you in touch with people that may have already cleared the tissue that you're interested in. One example data set that we have, this should be a video, but doesn't look like it wants to play. Let me just turn off my laser pointer. This is a guinea pig cochlear, which has been chemically cleared. That process takes about 3 weeks because this is primarily made up of bone. So decalcification is quite a long step. And here we can see the spiral within the cochlear. This group's actually looking at cochlear implants and looking at regions of damage caused by cochlear implants to see if they can improve the outcomes of those surgeries. All right, let's move on to multiphoton. So the light sheet in our example for large tissue is very much a fixed sample imaging approach. With multiphoton microscopy, we can actually do what we call intravital imaging on live anesthetized animals. And what we're doing here, if you remember the Jablonski diagram that we had from the last lecture, rather than having a single photon of light take us from the ground state up to the excited state, what we're actually looking is having, in this case, two photons hitting the molecule at the same time and causing the same effect. So in this case, we use longer wavelengths of light because they can penetrate into the tissue more deeply with fewer scattering effects, but we still get the same excitation as we would do for a single photon technique. So in this approach, we're pulsing photons onto the sample in the hope that, as I say, two photons will hit the molecule at the same time and cause this excitation effect. This means that to excite GFP, we'd normally be doing that with single photon at about 490 nanometers. With two photon excitation, we're using something more like 920 nanometers to get this two photon effect. And show you one example of this. This is one experiment looking at different T-cell immune cells within skin responding to a herpes infection. So in this case, we've got collagen in blue. In green is our herpes-specific T cells. In red is our non-herpes-specific T cells. And in magenta pink, we've got the herpes. And in this case, yeah, the group here from microbiology and immunology can quantify the motion of these T cells and see if there are different behaviors between the two different subsets. So very good for imaging, a few hundred microns up to potentially a mil into live samples, if that's what you're interested in. Next approach that we're going to look at is the slide scanner. This, again, is very much a fixed sample approach. So in this case, we would be taking either the paraffin or cryopreservation approach of fixing, embedding, and sectioning onto slides. The power on this system is that it is high throughput. So we can load in, this is our slide scanner, the AxioScan 7. We can load in 100 slides at a time onto the system, and we can get it to semi-automatically find the tissue and image whatever channels we want. We can do bright field and fluorescence imaging on here. We've got ranging from 5 up to 40x objectives on that system. And again, this is kind of meant to be plug and play. So you come in, set it up, press go, and then leave it to scan overnight. In this case, we have our preview scan showing where the tissues are. We can go in and define that. So we can use bright field or fluorescence for that. Here we have a few different examples of an overview scan, the same region at 20x and also at 40x as well. So again, it's not going to get you fantastic subcellular resolution, but if you're trying to count or visualize structures, then this is a much faster way of doing it than doing it manually on an epifluorescence scope or on a confocal microscope. Next thing we're going to look at is high content imaging. So similar to the slide scanner, this is high throughput and automated imaging. But in this case, we're not imaging samples on a slide or in a dish. We're actually going to mount samples into some kind of microtiter plate. So our system, which is the operetta, can take anywhere from 6-well plates up to 384-well plates. And essentially, if you can stain your samples and specific regions of interest, we can set the system to automatically image each of these wells for you with multiple fields of views if you need. The real power on this system is the automated quantification. So you're going to generate thousands of images potentially from this plate. You don't have to be manually going through and counting each of those. So we spend quite a bit of time with the researchers going through and optimizing their quantitative workflows so that at the end of the day, you get a heat map or an Excel spreadsheet or a graph of what your data is rather than having manually to go through and do the analysis yourself. So the pros are automation, unbiased imaging, high throughput. We can do assay validation and screening. Lots of people here are doing like drug studies or multiple different treatments to try and look at combinations that work. Downside is it takes quite a bit of time to optimize. So sample prep is key. The image analysis workflow is key. It can generate huge amounts of data. And yeah, it's not specific for single regions of interest. We can't define this position in this well and this position in that well. It's just going to image the same region in each of the wells that you tell it to. Having said that, the possible applications are pretty endless. This is a few that we've done. So looking at migration assays, this is, I guess, a variation of the scratch assay that some people may have done, where there's a plug which you can remove and image over time and look for the movement of cells into the plug. We've done experiments over time where we can track cells, again, looking at kind of chemotaxis type experiments. Or we can look at morphological analysis. So, what happens to cell shape after different treatments, gene knockouts, drug treatments, that type of thing. There's a really long, large list of things on the left-hand side there that you could do with these high content screening systems. Next thing I wanted to cover was wide field. So we're going down in the scales and kind of getting more to the cellular level now. So we went through a wide field or epifluorescence microscope last week, and we touched a little bit on image deconvolution. So using a mathematical formula to reassign the out of focus light back to its starting point. This is really ideal for quite thin samples, so anything that's less than 20 microns. Things like cultured mammalian cells. The example shown here is a malaria parasite imaged in wide field mode in 3D, and then using that 3D information to deconvolve and create a kind of pseudo confocal quality image. As I mentioned, what we're doing is a bit of mathematics to restore back the fluorescence to its original starting point. We use what's called the point spread function of the microscope to do that. That's going to give us an increased resolution in all 3 dimensions, increased contrast, and also remove the noise as well. So you can see here, raw image and then deconvolved image on the right. What that requires is ideally Nyquist sampling. So we have these Huygens licenses, and if you're on a 60x objective with a 1.4 NA, then they ask that your pixel sizes are about 50 nanometers for this to work. So you want really good quality sampling. And also we need 3-dimensional information to assign that light back to its original focal plane. The Huygens licenses, which we have here at BOMP, have a wizard, which will take you through how to do this on wide field confocal area scan and light sheet data. That has both GPU acceleration to allow that to go faster, and also batch processing as well. So if you're doing the same thing over and over to multiple images, you can set it up and just leave it to run. This is, I guess, an overview of the process. If this is what our light looks like, by the time it's gone through the microscope, it's a bit bigger and fuzzier just because of the way the optics have worked. What we're trying to do is use maths to bring it back to this original point. Okay, so that's everything I wanted to cover in your kind of up to cellular-based resolution. The next few applications that we'll go through will focus on cells, organelles, and proteins. And we're going to cover a few applications here again. So turf microscopy, confocal microscopy, a group of methods called the F experiments. And then we'll go into some of the super resolution methods as well. So confocal, super resolution, structured illumination microscopy, and then single molecule localization microscopy. So turf microscopy, which is also known as total internal reflection microscopy. So for standard wide field confocal imaging, we take so this example shows wide field. We're illuminating our entire field of view, our entire volume with the entire light coming from the entire objective. In turf microscopy, what we do is collimate the beam so it's a bit tighter, and we can translate it across to one side of the objective. What that allows us to do is to change the angle that that light comes out of the objective. So for this wide field example, we have translation straight through. If we get the angle just right, then we can get this total reflection. So actually the majority of the fluorescence will bounce off the cover slip and reflect away. The benefit this has is that we're not illuminating the majority of our sample with the laser. What we get is something called an evanescence field, which will just illuminate the first 100 nanometers past our cover slip. Okay, so we're just seeing the structures within that first 100 nanometers into our sample, which in some ways limits the application of turf microscopy. But it can be very good for high contrast live cell imaging and some super resolution techniques where we don't want to illuminate and potentially cause photobleaching or phototoxicity on our samples. We just want to illuminate this very thin optical section at the cover slip interface. And you can see an example here of an image taken in epifluorescence mode, not very much contrast, versus the turf mode, which is high contrast and also more gentle on our sample. So we have a couple of instruments available within our platform and a neighboring platform, which allow us to do that type of turf imaging. All right, so what we're going to do next is go through confocal microscopy. Now, obviously, there's a lot of different applications in confocal microscopy. We can't go through them all, but we'll go through some of those. First thing that's can become really important is the automated stage function on these microscopes. So that allows us to do 3D data acquisition. It also allows us to do multi-point visiting experiments and or tile scanning experiments as well. And I'll show you an example of that in the next slide. We can also do dynamic imaging, so imaging over time, looking at live cell imaging. Those F experiments will come back to looking at protein dynamics and protein-protein interactions. We can also do photoconversion experiments. So we discussed a little bit, I think, around the photoconvertible or photoswitchable fluorescent proteins in lecture 1. And then finally, we have specialized detectors on there again, potentially that can allow super resolution microscopy or spectral detection of fluorescence. First thing that we're going to look at is the automated stage functions. So in this case, again, this is data from the University of Melbourne. This is a retina, so the back part of your eye. And what the researchers here do is to take a whole map. So they cut the eye in half. They make 4 little incisions on that cup to allow them to make up what we call a flat mount. So this will sit flat underneath a slide and a cover slip. What they want to do then is get some really relatively high resolution imaging, but do that across the entire tissue. So if you took in one field of view, this entire sample, you wouldn't get very high resolution, high quality data. What they do here is make use of the automated stage. So if this is a single field of view, it will take that image, move the sample across, take another image, move the sample across. This is what we call a tile scan. That's what's happening on the slide scan system. We can then stitch those separate images together into making a much larger image. I mentioned around specialist detectors. So one example here is spectral imaging. So here we can acquire data of multiple fluorophores, and in this case, this is an example of a researcher with 9 separate fluorophores on their sample. What they've had to do in this case is take individual color controls to allow us to measure what the spectra look like on the microscopes. We can then use a mixing software to separate out those 9 different stains. Technically on the microscope, there's a couple of different ways of doing this. So in this case, we've got a diffraction grating. Sometimes it can be a prism as well, which separates the light out into the different wavelengths. And then in front of our detector here, which is the PMT tube, we have this variable slit. So we can actually vary what wavelengths go through to our detector. Alternatively, there are a few systems out there again with a diffraction grating to separate out into the different wavelengths. And this is spread across a multi-channel detector. So in this case, this would be something like a 32-channel array, which allows us to capture each or kind of separate out each of these wavelengths into separate bins. The benefit here is it's a single acquisition. For the example at the top, we need to move this slit across over time, so it takes a bit of time to acquire the data. But certainly you can see this can be very, very useful in terms of separating out multiple stains. And we have one system now which can unmix up to 15 stains, if that's something that's of interest to you. Up till now, we've been talking primarily about point scanning confocals. So if you remember, using mirrors to place the laser and build the image up over time by scanning that laser across our sample. There is another form of confocal as well, which is called a spinning disk confocal. In this case, as the name suggests, we've got a disk which spins, and in that there are lots and lots of little pinholes. So rather than having a single pinhole in front of the detector, the spinning disk has all the pinholes sat on the disk. This has the benefit that it's much, much faster acquisition time. So the readout here is actually onto a camera rather than onto a point detector. So here we're not scanning across, we're spinning this disk around, illuminating just a small part of the sample at any one time. The downside is often with thicker samples, it's a little bit harder because you can get crosstalk between each of the pinholes on the disk, which deteriorates your image quality. Just go back. Yeah. This is an example of some data taken on a spinning disk system. So here we're looking at mitosis. If you're ever trying to image mitosis, then it can be quite sensitive. So often if you put too much light on there, you will find that your cells will hit a checkpoint and just stop replicating. So yeah, spinning disk confocal, very fast acquisition, nice for clearer 3D samples. Or for fast live cell imaging as well. All right, I also mentioned protein diffusion and interactions. We've mentioned the F experiments, which we're kind of grouping together based on the first letter of the acronym. So there's a few of those which we'll go through over the next few slides in detail. So the first one we want to look at is something called fluorescence recovery after photobleaching. That's shortened to FRAP. The next one after that is FLIP. Then we have fluorescence fluctuation spectroscopy. FLIP is fluorescence loss in photobleaching. Fluorescence fluctuation spectroscopy. And then we have fluorescence, or sometimes called Forster resonance energy transfer. I touched a little bit on that in lecture 1, but we'll go through it again in this lecture. So the first one is fluorescence recovery after photobleaching. In this case, and we've got an example images here, what we're looking for is the mobility of the molecules within this red region here, or within the sample. What we're going to do is to use high-powered lasers. So this is normally laser scanning confocal. We're going to increase the laser intensity here to actually selectively bleach out the fluorescence within this red box. We will then image over time, and what we're looking for is the recovery of fluorescence into that red region. So what we're looking at is mobility of those molecules within your sample. If we look at the figure here on the right-hand side, what we're looking at is, sorry, my mouse is being very glitchy. We're looking at the intensity within that box, within the red box. So to start with, you can see that's level. struggling to get my mouse to go where I want it to go. We can see that's level at the start. We're imaging over time, so there's not much photo bleaching happening. We then increase the laser. You can see the big drop there in intensity down to the bottom. And then over time, you can see the recovery of fluorescence back into that red box. So we've got a couple of things here. We can measure the half-time of the recovery, so how fast that recovery is happening. But we can also quantify the percentage of the mobile and immobile fraction of those molecules within your sample. Some example applications here are, for example, looking at membrane or membrane protein dynamics. So here we've got syntaxin 1, which is selectively bleached, and then we're looking at recovery over time. Same thing with the SNAP25. We can look at diffusion of proteins across structures. So in this case, we're looking at diffusion through the nuclear pore complex. So we're bleaching out the nucleus and looking at recovery of molecules coming from the cytoplasm into the nucleus. Again, we can quantify those using those approaches. A related method is called fluorescence loss in photobleaching. In this case, we have one region of interest, but we're doing multiple bleaches within that region of interest. So in this case, we've got our pre-bleach. We're selecting our region here that we're wanting to bleach out, and we keep bleaching that region. What we're looking at is the connectivity, the continuity between the other parts of the cell with the region that we're bleaching out. Yeah, so over time, you can see the cytoplasm is highly connected with this region. And over time, the membrane does start to lose intensity. So it does suggest that it is connected in some way. Fluorescence fluctuation spectroscopy. So these are a group of methods. And again, these are highly complex. You could probably do at least an hour on just these. Sometimes called things like FCS, for example. In this case, we can use the confocal again, and rather than scanning across and building up our image, we can park the laser beam at one specific part of the cell. And what we're trying to do is measure the intensity within that volume over time. So as our molecules come in and out of that excitation volume, we will see the intensity go up. What we can do is then fit that curve to try and work out what the diffusion coefficients of the molecules are. Things like, are they oligomeric, monomeric, that type of thing. If we combine that with what we call raster scanning, so your standard confocal scanning across the sample, we can also do analysis like number and brightness analysis, which again will give you an idea of the oligomeric state of your sample, but on a whole image rather than just on a single volume of your sample. Last one that we'll spend quite a bit of time on is the FRET applications of fluorescence resonance energy transfer, or sometimes called Forster resonance energy transfer. We did touch on this in the earlier lecture, I think. This is a way of looking at protein-protein interactions. Here we have one molecule that we're trying to work out if it interacts with its substrate. In this case, we've attached the sensory domain here to a CFP protein, and the substrate is attached to the YFP protein. What we're trying to look at is interaction of the substrate and the sensory domain. So when there is no interaction, if we excite the CFP molecule, we can see the CFP molecule emitting. That's at this 475 nanometer. Sorry, just move my zoom thing. Up here at 475 nanometers. When these two molecules interact, that's going to bring the two fluorescent proteins together. They need to be very close, so within 10 nanometer of each other for the energy transfer to occur. So in this case, when they are within that 10 nanometers, if we excite the CFP molecule, rather than the CFP molecule emitting the light, it actually transfers that energy to the YFP molecule, exciting the YFP molecule, and therefore we have emission coming from the YFP molecule rather than the CFP molecule. And if we look at the emission curves here, you can see that that's shifted to the YFP species from the CFP species. So again, in terms of FRET setup, we can look at the Jablonski diagrams again if we want to. So here we've got the excitation of our CFP molecule. Rather than losing some energy and going back down to the ground state, that energy is transferred to the YFP molecule, meaning it goes into the excited state, can go through the normal energy transfers before going down to the ground state and emitting that molecule. One of the key things when designing your experiments for FRET is that you want to make sure that the emission curve of your donor, in this case CFP, overlaps quite a large bit with the excitation curve of your acceptor, which is the YFP molecule here. So this area in green you want to be as large as possible for most efficient transfer of the energy. What are some of the applications we can do here? So I mentioned things like protein-protein interactions. So if you have two proteins you think interact, and they're both tagged with these fluorescent proteins, if they're close enough, then you can read that out. There are things like calcium sensing. So here we've got a molecule which, when calcium binds, it causes a structural rearrangement of the molecule, bringing these two fluorophores within that FRET pair distance. Kinase activity. So here again, when we have phosphorylation, this again causes a structural rearrangement, producing FRET. And then we also potentially have protease activity here as well. So if we have a protease happening, it's separating out and reducing the FRET efficiency on these in these cases. FRET analysis can be done in a number of ways. And again, I would encourage you to reach out before you start these to get some advice. So we have systems which can have workflows which can help you through this. So one method is acceptor photobleaching, in which case we're bleaching out the acceptor, in this case on the right-hand side. That should increase the intensity coming from the donor. We can do ratiometric FRET. That's only for FRET pairs with a fixed stoichiometry. So in those previous examples, those molecules which have a structural rearrangement always have a one-to-one stoichiometry. So we can do ratiometric here. There's a method called sensitized emission, which requires a lot of controls and a lot of mathematics. But probably the cleanest way of doing it is to use something called fluorescence lifetime. This is nice thing here is independent of the wavelengths of the light that we're using. It relies on the fluorescence lifetime. So what I'll go through next is the fluorescence lifetime. So if you remember, that's the length of time that the molecules will stay in that excited state. And that is very different for each of the molecules. Example in this table on the right-hand side here, if we have, say, elixafluor 488 and enhanced GFP, spectrally those two would sit directly on top of each other, but they have very different lifetimes. So elixafluor 488's lifetime is about 4.1 nanoseconds, GFP is 2.6, so we can use that to spectrally separate those out. So I mentioned fluorescence lifetime can be very useful for FLIM-FRET applications. We can also use it for multispectral analysis. So again, trying to separate out multiple fluorophores. We can also use it for autofluorescence imaging, so things like imaging NADH, NADPH, that type of readout. In this case, we excite our samples with pulses, and what we're looking at is to measure the time difference to when we actually collect the photon on the detector. And this difference we can read out as a proxy for the lifetime of the fluorophore. Here we can plot this, and we can fit a curve to this, which will tell us how many species there are within the samples. This is an example of a plant sample, which has been imaged with a film microscope, and it's showing very different spectral lifetimes throughout the sample. One way of analyzing FRET data is to use what's called a phasor plot. This is a way of visually separating out the FRET signal. In this case, we've got histone T2b, which is tagged with either GFP or mCherry. When the histones are fairly well separated, you get no frets, and as they become more condensed, you can see you can increase frets. In the phasor plot, we can spatially separate these on the plots to be able to quantify the different levels of interaction of those fret molecules. All right, so that was the F experiments. So now we're moving into the kind of super resolution microscopy space. So if you remember, I was talking about the diffraction limited resolution of standard microscopy is kind of in the 200 to 250 nanometer direction in the XY direction, and about 600 nanometers in the Z direction. There are a group of super resolution microscopy techniques which are available, which can get around that diffraction limit, and they do that in multiple different ways. We will go through some of those that are available at the University of Melbourne for you. Some of these come with caveats. So in this table here, I've shown not just the resolution that we can achieve in X, Y, and Z, but also the imaging depth that we can image these samples at. So as I say, for confocal, we're about 250 nanometers XY, 600 nanometers in Z, and your best imaging is going to happen in the first 50 microns past the cover slip. One of the techniques we'll go through in the next slide is AEST. That's a confocal-based technique. Again, gives you an increase in resolution without much issues in terms of depth penetration. As you see, as we go down through the increasing the resolution, you can see that often we are sacrificing some of the depth that we can image our samples into. All right, so first technique that I wanted to cover was confocal super resolution, and at the University of Melbourne, the technique we use for this is something called AEST. I'm aware that Nikon also have the NSpark detector that works in a similar way. So in this case, it's an additional detector which we use. So if you remember, standard confocal imaging is a single point detector. So yeah, as we're scanning across, it's reading back to that single point detector. For the AEST detector, we're using this slightly strange setup with a 32 detector array in this kind of hexagonal grid. And the reason we're doing this is that we know in standard confocal setup that we normally have the pinhole at 1 area unit. If we want to increase resolution on the standard confocal, we can narrow the pinhole down. What AEST does is actually keeps the pinhole at 1.25 area units, so actually widens out the pinhole a little bit. But what we're doing is we're separating out the detection into each one of these individual detectors, and each individual detector is equivalent of 0.2 of an area unit. So we're not sacrificing anything in terms of the light going onto the system, but we are able to sample much smaller pinhole sizes with what we call pixel reassignment. So there's a bit of mathematics post-processing that goes into this. It will get us a 1.7-fold increase in resolution in 3 directions. Some examples here, again from the University of Melbourne, is confocal data sets versus AEST and AEST process data sets. And again, for some of these lower intensity regions, we can start to see some much higher resolution information than we are able to on a standard confocal data sets. I did mention earlier on that we had a Huygens deconvolution package. So often if we're trying to get the best resolution out of area scan data, we will run it through the Huygens deconvolution package. You can see an example on the top here of data set, which is acquired with area scan processing versus the Huygens deconvolution, which again provides that increased signal to noise, increased contrast on our image, and therefore provides a little bit extra resolution improvement. Structural illumination, so specifically 3D structural illumination. This can provide up to an 8-fold increase in resolution in X, Y, and Z. So 2-fold in X, 2-fold in Y, 2-fold in Z. Here we can do 4-color imaging. The downside is we need to take more exposures than we would do on a standard confocal system to be able to acquire that data set. So what we do is we image through an interference pattern that interacts with our sample and provides us with deeper a higher resolution information, which we can use to, again, post-process and compute the improved image quality. Here we've got some examples. This is Hendra virus imaged in wide field mode on the left and 3D sim mode on the right. These are the Hendra coat proteins. It's about 300 nanometers in diameter, this virus. This again is the malaria parasite that I showed earlier in the talk in wide field mode versus 3D structural illumination mode. And again, these little protein aggregates here, these little puncta are about 100 nanometers in diameter. And then finally, we've got a eukaryotic cell where we're showing a tubulin in wide field mode or 3D sim mode on the right-hand side. Last super resolution technique that we'll go through is single molecule localization microscopy. This is a group of different techniques. So they all work in a similar way in terms of the post-processing. So here we have essentially a movie of the Eiffel Tower. Go back through and play it. So it blinks at night. What we're trying to do with these techniques is to make sure that most of the fluorophores aren't emitting at any one time. And we're looking at just a few fluorophores in the sample emitting. That allows us to then, with certainty, be able to say that's a single fluorophore. I'm going to mark that position. And then we take thousands of frames to build up that super resolution microscopy image here. You can see from here, the ring here, the lights don't blink. And therefore they create gaps in the analysis because they're excluded from the analysis. Question is, how do we get our fluorophores to blink? Because normally in fluorescence microscopy, we want all those molecules emitting the light back out. There are lots of different ways of doing this. 3 of the most common ones. One is PALM, so photoactivated localization microscopy. Here we're using specialist fluorescent proteins normally to where we can control using either photo switching or photo activation when those molecules are emitting. So we're in the case of photo switching, we can use an ultraviolet laser to convert a few molecules from emitting from a green into a red wavelength, and we measure those emissions coming through in the red wavelengths as the blinks. For storm, these are chemical, normally antibody-based dyes. Again, we're using high reducing power, high laser power to push most of those fluorophores into a long-lived off state. Only a few of those will return down to the ground state so they can go through fluorescence and emit, and we get that blinking effect that way. With DNA paint, this is point accumulation for imaging in nanoscale topography. Here we've got our antibody that we're interested in, which has an oligonucleotide attached to it. We would go in with a complementary oligonucleotide with a fluorophore attached to it, and we're looking at the stochastic binding and release of those oligonucleotides. So that is what we use to build up that blinking effect. This again is an example of, in this case, D-storm experiments. Again, wide field imaging on the left-hand side, and then the single molecule imaging on the right-hand side. These are those 100 nanometer structures we saw in 3D Sim before, which we can start to now see some substructure in single molecule. All right, so we've been through quite a lot, and apologies for not sharing the screens on the first half of that. As I say, I'm happy to share the slides so you can go back through that content. So hopefully you can see how we can use fluorescence microscopy approaches to image samples all the way from organs, small model organisms, through tissues, through cells, through organelles, down into kind of molecular level once we go past that diffraction limit. As I mentioned, once we get beyond the kind of 20 nanometer resolution, then we need to move into some of the electron microscopy applications. All right, so last section that we're going to go through is ethics in imaging. So once you have generated your data, what should you be doing with it? So the first thing I want to talk a little bit about is data management and how you should be handling that data once you've acquired it on the microscopes. So first rule that we always suggest to our users is always save that in your original raw file extension. So depending on what company you're using, the file extensions change. Try not to, if you can, save this data as a TIFF or a JPEG file. The reason we suggest that is these files don't just save the image, but they also save the metadata as well. So exactly how that image was acquired. I don't see any of our researchers sitting there with their lab books making notes of what their zoom value and what the laser setting was and what the detector gain setting was. When you come to publish, it's becoming more and more prevalent that you need to provide that type of data with your raw data for the publication. The other benefit you often have in the software is you can use that old file to reassign and reapply those settings. So that makes your life a lot easier down the track as well. So once you've got your raw file, if you do want to, convert it to something that's a little bit more manageable, then if you are going to export it, then we suggest that you do it into a .tiff file. So this is what we call a lossless compression. So we're not losing any information. The file size is large. It should be used for data analysis and publication. If you're converting to a JPEG, a GIF, or a PNG file, this is often a lossy compression. Your file size is small. You can use it for presentation, but ideally not for any data analysis. So if I take a TIFF copy of an image and I subtract the JPEG version of that image, you can see the data that's actually been lost in the conversion to JPEG. Once you have started to work on a file, I always tend to get in the habit of trying to obviously never modifying that original file. If you are going to make any manipulations to it, then I would copy that across. So you've got image one. If you're doing different processes, then you should annotate what those processes are within the file name, ideally. Next thing, and again, this becomes really important, we have some horror stories here where people have been carrying their entire PhD's worth of imaging around on USB drives and have dropped them and lost them and things like that. In this day and age of cloud compute, that shouldn't be an issue to any of you guys, but something for you to be aware of. So yeah, always keep the original data file safe and unchanged. Please double back up your data. So this is an agreement that your supervisor makes with their funding body that they're going to handle that data correctly. If you're not sure what those rules are, maybe check in with your supervisor. And certainly within our facility, we recommend that you don't try and use any of our microscope computers as a safe place to back up your data. They have multiple people that can access them and could go in and change things or just delete the files completely. So once you've got any original data modified, you've made note of your modification process, you've got quantified results, ideally we want you to double back that up. And if you're within the university, you should have access to a research drive or a media flux project if you've requested one, which has that disaster recovery. So if there's a fire in the data center, the university has a backup copy of that elsewhere. The other thing to think about is that, although we're creating an image, this is still scientific data, right? So if we're making a conversion from an original image into, a modified image, we need to still be doing that in an ethical manner. And we're going to cover that over the next few slides, and make it kind of clear to you what is okay and what is not okay. So there's a number of papers out there which cover this topic. Again, these slides will be available to you if you want to go and read through any of those. But yeah, we're going to go through some examples at the moment in terms of modifications that have been made. So here we've got our original image here. In this case, they've removed this band over here. And well, they added some extra bands in here. Okay, so hopefully you guys can see that that's not ethical. If we are bringing in this case from Western blots, from separate blots, then it should be separated out clearly and made obvious that these are not from the same original data set. That's Western blots. We're here to talk about microscopy. In this example, we have got an electron microscopy image here. In electron microscopy, you can label proteins of interest in your sample with antibodies, and those antibodies are conjugated to gold particles. The idea being that the gold absorbs the electrons and show up quite strongly in the image. In the raw image on the left-hand side here, you can see we've got this big dark object here, which presumably is non-specific stain because the researchers have gone in and taken that out. They've also gone in and enhanced these objects here. Should have said with the immunogold, you can have them in different sizes, so you can do multiple labeling experiments. In this case, they must think that these are their gold particles, labeling the structure that they're interested in because they've gone in and modified the intensity of those objects. So in this case, again, this is unethical. They're going in and making specific changes to specific parts of the image. Finally, we're getting a little bit stronger. This is a light microscope image. In this case, we've got this fold in the tissue here, which they've gone in and taken out. So they've tried to clear that up post-imaging. Again, this is unethical. You're making a specific change to one part of the image. What they could have done here is, leave it. If the fold in the tissue is not adversely affecting the analysis of the data, so you could just leave it in there. If it's not affecting analysis of the data, you could also potentially crop it out. Ideally, what you should probably do is go back to the block that you've got, resection that, staining it, and imaging it without the fold sitting in the image. Next example is a fluorescence microscope image. So this looks fairly good at the moment. If we go in and make a very simple contrast adjustment, then you can see that these groups of cells here and these groups of cells here have a very different background to the main image, which suggests that they might have been copied in from elsewhere. So again, this is clearly unethical, making these types of modifications within images. And the difficulty now is the journal editors, when you submit a paper, will go through your data forensically like this and try and look for any of these problems. So what modifications can we make? So here we can, if we've got an image here, maybe we haven't acquired it very well. We've not used very much of the dynamic range on the detector. So this is an 8-bit scale from 0 to 255 grayscale values. We're down at probably 30 intensity values here, so it makes it difficult to visualize the data. We can adjust the brightness here to make that a little bit more visible, a little easier to see. Key thing is that we need to say that that's what we've done within the image file or within the legend in your publication. What we don't want to do is go in and increase the brightness again until we've gone past where our dynamic range is. So here we're saturating these pixels. We can't quantify it. You'll notice the structures look way bigger than they actually are in the actual data. So that's again something we want to try and avoid, even if we are making a modification to the whole image. This is another example of a fluorescence image. So in this case, it looks good. We've got a negative, a positive, and a sample here. Presumably they're trying to compare intensity values across these three different data sets. The problem is, again, if you come in and do a contrast adjustment, that the background is very different in the negative control, the positive control, and the sample, which means either they've acquired the data differently or they've handled the data differently when they're presenting it. That raises a few questions in terms of how they've actually analyzed their data here. So ideally, if you want to directly compare data, it should all be acquired in exactly the same way. Another example here is, again, a bright field image. Here they've removed this background tissue from this data. And they've also evened up the illumination of the sample as well. In this case, we'd say that's unethical because there's a large degree of background alteration, even if you're clarified within the figure legend. Oops. Last one is modification on the background. So in this example, we've got a bit of a shading issue on the right-hand side here, which has been cleared out. Normally, I would say here that you're better to make sure your microscope's aligned properly and capture the data nicely rather than trying to fix it afterwards. This would be acceptable only if no change has occurred during the actual tissue. And again, you've made a statement in the figure legend that you've done that, but yeah, ideally you want to capture that data correctly. What are we trying to do when we're acquiring this data? And for me, what that is we should be trying to capture data that looks the same as what we see down the eyepiece. So I always say that 80% of the battle in microscopy is sample prep. If you get a good sample, then the imaging becomes easier. The analysis side becomes easier as well. If you put, a faint sample in, then you should get a faint sample out. What we want to try and avoid is that you tweak your settings on the microscope to actually make something that's poorly stained actually look halfway decent. Again, that's not really an ethical practice. All right, so best practice with your image acquisition, any signal can be amplified or suppressed to look like positive or negative signal. Ideally, we want to calibrate that data to a known standard. So certainly if you're doing intensity-based readouts, I normally suggest you have an internal control within your experiment that you can normalize to. Ideally, you want identical acquisition settings between different samples if you want to be able to directly compare them. We want to have identical post-processing between those samples as well. You should be retaining the original files for post-assessment if possible, and ideally documenting all the processes that you've done. So you should be able to tell someone what you've done at each of those steps. So that's the end of lecture 2. So we have gone through sample preparation, general advice. We've gone through quite a lot of different experimental applications that are possible on fluorescence microscopes. And we've also gone a little bit through imaging ethics and what to do with our digital data once we've collected it. As I say, next week is the last webinar in the series. So my colleague Ellie Cho, who is our image analyst, is going to go through fundamentals of bioimage analysis. She'll take you through the workflow of things to consider from experimental acquisition, pre-processing of the data, how to identify the structures that you're interested in, and what readouts you can make out of that. So yeah, hopefully you guys can come along to that next week. Otherwise, I will leave it there with this slides up, and I will start to dig through the Q&A.